Good morning or good afternoon. I'd like to welcome you to the Biochemical Society in Portland Press Webinars Focus Series, Biochemistry Focus Webinar Series. The topics in this series include molecular biosciences as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text. We are welcoming suggestions for other webinars as well as suggested speakers for future webinar series. Please visit our website for more details. My name is Deborah Schechtman. I am an associate professor at the University of São Paulo, Brazil, and the Department of Biochemistry, and an associate editor at Bioscience Reports. Today's webinar is featuring a special issue of Bioscience Reports on imbalance of homeostasis in the nervous system. We are currently inviting content for the special issues, new insights into homeostasis and strategies for therapeutic strategies. Today's session, we will hear from Professor Alberto Javier Ramos and Professor Kathryn. They will discuss the role of glial cells and neuron-glial interactions and their influence in molecular mechanisms in neurological diseases. Before I hand in to our first speaker, I'd like to mention that you are welcome to ask questions, sending your questions during the talk just by writing it in the question box in the upper right-hand corner in the screen. Then in the end of our session, we will be answering your questions. So I'd like to welcome our first speaker today. It is a pleasure to invite Professor Dr. Alberto Javier Ramos from the University of Buenos Aires. Alberto obtained his bachelor degree in biochemistry and PhD at the University of Buenos Aires. Then he did 2 postdoctoral fellowships, one at Austral University in Argentina and the other in Montreal at the Neurological Institute in the Hospital of McGill University, Canada. Today, he will talk to us about glial response in the central nervous system and injuries. And in epileptogenesis and its treatments. Thank you, Javier.
To you and the organizers and the Biochemistry Focus webinars for inviting me. We recently published this paper that you are seeing the title in my talk, and I will try to let you know what we found and why we think it's very development of the epilepsy and the possibilities of treatment. So if you are seeing okay my slides, I will go. So epilepsy has been with us for a lot of time. From 4,000 years, we have been together with epilepsy in the world. This is an old Babylonian script. When they describe 1,000 years before Christ, they describe the clinical features of epilepsy with details and the seizures and how to treat those patients. They suppose that was a problem of a devil inside the patient at that time. But anyway, the description is really clinical. And this is the Inca king seeing his wife having a seizure, also in very early time in America. So epilepsy has been with us, with the human beings, for a long time. Now we have 50 million patients worldwide. We have almost 70 cases, new cases per 100,000 inhabitants per year. This is in a city of 1 million people. It means that you have 700 new cases. That means 2 cases per day. It's really incredible, the number of patients that we have with epilepsy. And the problem is that nearly 80% of the people with epilepsy, the patients with epilepsy, live in low and middle income countries. And that's even worse because the treatment is not usually available for all of them. The more important risk is the premature death in the people with epilepsy, because it's 3 times higher than in the general populations. And we have available more than 30 anti-seizure medication, but they are not anti-epileptic. They just treat the seizures, but they don't stop the development of the disease. And in the scenario is even worse because 30% of the patients with epilepsy, in the long term, they probably develop drug-resistant epilepsy, so they fail with several treatments, and there are no much options for them. The only thing that we can offer in the clinical is the surgery, the resection of the epileptic focus, and sometimes devices like vagal stimulation or deep brain stimulation can be used, but they are not usually useful in all of the patients. So these patients with drugs with sudden death in a SADEP syndrome, that's the higher risk, and they have cognitive and physical impairment and social impact because they basically can't work and behaving like the rest of the people. So the problem is really big. The anti-seizure medications that we have available on the clinical grounds are basically dedicated to treat excitatory synapses or inhibitory synapses, because the theory of beyond epilepsy is that it's an excitatory inhibitory disbalance in the brain. So the balance is wrong, and so most of the anti-seizure drugs, they work on the excitatory synapses or inhibitory synapses. Even the cannabidiol, which is maybe the most famous anti-epileptic drug developed in the last years, is also acting on this type of synapses. But we know that neurons are not alone. Neurons are together in the brain with astrocytes, which is the focus of my paper and the work of my lab, and oligodendrocytes and microglia. So, and this is a blood vessel that is, also a participant of this story. So basically, neurons are not alone, and that's one of the problems in epilepsy, because most of the medication have been dedicated to neurons, but didn't pay attention to the rest of the players in this field. Astroglial functions in the normal brain are many. For example, they provide lactate to neurons. Neurons depend on the lactate. They use the lactate provided by astrocytes to support the metabolism. Astrocytes also are a glycogen storage in the brain. They keep the ionic homeostasis. For example, they reduce the level of potassium in the extracellular side, reducing the excitability of the neurons. They uptake neurotransmitter, including glutamate, preventing excitotoxicity. They secrete gliotransmitter. They couple neuronal activity with local blood flow, and they provide factors, trophic factors to neurons. They also crosstalk with the professional immune cells. My lab has been working in this stuff for a long time, describing how astrocytes interact with microglia, with lymphocytes, and even with the peripheral organs like the spleen. And astrocytes are also essential for blood-brain barrier formation and maintenance. So they are really important cells in the brain. This is a picture of my, from my lab, showing a blood vessel and how astrocytes cover the entire surface of the blood vessel, making the blood-brain barrier. Epilepsy doesn't start as epilepsy. Epilepsy as a chronic disease starts many years before, usually by one initial lesion, or we call that initial precipitating event. Could be status epilepticus in the young, childhood, a trauma, brain trauma, that could be also in adults, or in kids could be febrile seizures or encephalitis. This initial precipitating event sometimes induce seizures, but then stop. And for many years, from months to years, we have a silent situation where the patient is normal, but something is going on in their brains. For example, cellular plasticity, neurodegeneration, and structural reorganization of the brain. And that's one thing that we pay attention, is that in this patient, astrogliosis and microglia activation is produced. This is a silent period, and then after years, the patient start with chronic epilepsy. That means spontaneous seizures. So at this point, there are already, hyperexcitable networks and some dysregulation of glial cells that makes this system to keep on having seizures. So our underlying hypothesis on this work, from the clinical point of view, is that epileptogenesis is a long-term process, and then it's possible to modulate it to modify the natural story of the disease. So what we are thinking? We are thinking that epilepsy can be prevented if we intervene in clinical grounds, we intervene in this period of silent, development of the disease. Because in the moment that the patient is already chronic, the only thing that you can offer to them is the anti-seizure drugs that only prevent the seizures, but don't change the natural story of the disease. So our questions, based on this hypothesis, were are astrocytes remodeled during the epileptogenic period? Is this remodeling stable in time? Can facilitate by themselves epileptogenesis. And these remodeled astrocytes can be a potential therapeutic target. So the idea is to prevent epilepsy instead of only controlling chronic seizures. So the model that we use in animals in this paper we use several different models. We use in silico, in vivo, and in vitro experiments, and also material from humans. But the development of the disease in humans is an initial precipitating event, the silent period, and then the epilepsy. So in animals we can model that in short time in adult rats, in Wistar rats. Doing an status epilepticus induced by pilocarpine and lithium injection. And this is the initial precipitating event. Then we leave the animals to stay for 20 minutes having seizures. And then we stop the seizures with diazepam. And then we study the latency period where the animal is not epileptic yet, because the animal will be epileptic at 40 days, 40, 50 days after the initial precipitating event. So this latency period is modeling the latency period of the patients in human. The first experiment that we did, we've been working for like 10 or 15 years in astrocytes and this model. And these are images of the last paper, but we have a lot of data if you want to go back in time following the papers of the lab. But these are images that I took from these pictures just to illustrate you how this is a control astrocyte labeled with GFAP immunostaining. And these are astrocytes of an animal that suffered status epilepticus, and we wait for 7 days to see how they look. And this is 35 days after the initial precipitating event, and you can see that they are very different from normal astrocytes. So in this paper, that we published, what we did is to follow the 5-methyl cytosine methylation in the DNA, and we saw that there was an increase. This is a CA1 in the hippocampus, and you can see that this is stratum radiatum. And in this area, you can see all the methylated nucleus, and then the dramatic increase that we have after the seizures induced by lithium pilocarpine. When we analyze, but these are all cell types, but we did double immunostaining with GFA, and we analyzed how many of these, how much these astrocytes increase the methylation in their DNA. And you see here in the graphic and the quantification that there are many it's a most significant number of astrocytes showing an increase in 5-methyl cytosine in the DNA. And we know that also in these animals, we have neurodegeneration, microglial activation, lymphocyte infiltration in the brain, increases synaptic plasticity and spleen activation, etc. You can follow these papers if you wanna see, back in time all the story that we have been describing about this. So next experiment was to check by real-time PCR how is the expression of the complement C3, because this is a pro-inflammatory factor of astrocytes. And we did see that this increase at 7 days and 21 days after the initial precipitating event. But the interesting thing is that there was a decrease of the KIR4.1. This is a channel required for astrocyte to obtain and reduce neuronal excitability. So not having KIR4.1 in astrocyte means that they cannot prevent neuronal excitability, hyperexcitability, and this is very important in the epilepsy field. And also these are real times, like spots, like a temperature spots. And MAPG is a neurotoxic factor of astrocytes. GFAP, is a marker of ratty astrocytes. And the other interesting thing that we saw is that DNA methyltransferases number 3 and number 1 were increased after the initial precipitating event. These results, together with these results, started to make us to think in the lab that something was going on in those astrocytes, that it's not just a finding of one gene. So we check by immunohistochemistry the expression of aquaporin 4, a channel that is required for astrocyte to pump out water from the CNS, and glutamine synthetase, which is an enzyme that astrocytes require to transform the glutamate into glutamine, glutamate. And as you can see here, both those very important homeostatic genes were decreased after the initial precipitating event. So trying to understand what the mechanistic the mechanistic situation beyond these findings, we use the HNGB1. The HNGB1 is a damage-associated molecular pattern released by dying neurons and is recognized as a pro-epileptogenic factor. This has been published by many different groups around the world, and so it's very accepted in our field to use HNGB1 as a pro-epileptogenic factor in vitro. These are experiments in vitro, that's why we have a flash here. And you can see here astrocytes exposed to HNGB1, and this is the methylation, initial methylation. This is 24 hours, 22 hours after the exposure, and 7 days after the exposure. And as you see here, there is a nice increase of the methylation in the astrocytes in this culture after they are exposed to HNGB1. And this is the quantification just to this situation is very stable in time. One important thing that I'm sorry that this is in Spanish, I missed that, but anyway, this is an 18 hours exposure to HNGB1. Then the tissue culture is washed, and then we see the tissue culture during a long time, and you see that the methylation is very stable in time. 7 days for a cell in culture is a long time. So it seems that only just one exposure to HNGB1 keeps this astrocyte methylated for a long time. And then in the same culture, we analyze how is the expression of different genes, and we check lactate dehydrogenase. This is a transporter. The lactate dehydrogenase is an enzyme required to produce lactate to send it to the neurons. This is the MCT2 transporter that transports lactate into the neurons. This is glutamine synthase. I already talked about it. And this is the gene of the KIR4.1, the channel that reduces the potassium level in the extracellular site. And these are the cytokines, and again, the enzymes. And these 4 genes are homeostatic genes required for astrocytes to be nice with the neurons to provide nutrients to the neurons. These are pro-inflammatory factors, you know them, interleukin-1 beta and IL-6, interleukin-6. And this is the methylation enzymes for the DNA. And you see all the blue spots are due to, they are reduced after HNGB1 exposure. And again, this is a very, very stable situation in time. And all the red spots means that they are overexpressed. So we have a combination of homeostatic astrocytes with reduced capacity. These astrocytes are, not good for supporting neurons, but at the same time, they are pro-inflammatory, and they are overexpressing methylation enzymes. So that means that the situations looks more like a program of gene expression than a finding of a point situation in time. And this is just the real-time PCRs to show you that we have all the data, and the data is also in the paper. So thinking that this is a program, the next thing that we did is to go in silico and to check these promoters. I mean, the lactate dehydrogenase, the transporter of lactate, and the glutamine synthase promoters have CPG islands, which are candidates for being methylated. So this is in silico work. You can see the details in this paper that is was before the paper that I am commenting, the clinical science paper. But anyway, the data was super important to see that there are promoters that are methylated. But this is in silico. So the next thing that we did methylation specific PCR studies, And we saw that the methylation, in fact, is a really increase in these genes, in these promoters. So the following step was to try to do a loss of function study and a potential projection to the clinical grounds. We used the cytarabine, which is an FDA-approved DNA methyltransferase inhibitor, a pan-DNA methyltransferase inhibitor accepted for clinical use in cancers. And what we did was pre-exposure to DNA called to the cytarabine, and then we exposed the astrocyte to HMGB1, and then we analyzed what was going on. And as you can see here in this real-time PCR studies, the transporter, this is the lactate in astrocyte exposed to HMGB1, and this is the recovery of the normal expression when we use the cytarabine. And the same with lactate dehydrogenase, and the same with glutamine synthase, and with the transporter KIR4.1. The effect is not, again, it's not similar in all of them, but anyway, in all of the cases, we basically restore the expression, or we prevent the decrease in the expression of these homeostatic genes. And at the same time, we achieve a partial reduction in the pro-inflammatory factors. So it seems that we are interfering somehow with the program of astrocyte gene expression in this situation. If you are thinking, okay, but this is really working, this is an experiment that was not original in the paper, but we did it some time ago, and we saw that the methylation is reduced in astrocytes when you use the cytarabine. So this is, that is gene expression PCR, but it's also seeing the glutamine synthase is recovered when you use the cytarabine. You can see here the quantification. That we did was to use conditioned media of these astrocytes exposed to HMGB1 in presence or absence of the cytarabine, and we saw that the astrocytes, when you use the cytarabine, they don't kill neurons, but if you use vehicle instead of the cytarabine, they kill neurons. So it seems that astrocytes exposed to HMGB1 basically kill neurons. And if you use the cytarabine, they cannot kill neurons and they keep on supporting neurons. And this is just the image to show you. This is, is culture, tissue culture. This is in silico studies on rats, but what's going on in real patients. So the interesting thing with epilepsy is that you can get tissue from patients, because when they get surgery in epilepsy, in drug-resistant epilepsy, you have the material. So we took that material and we checked GFAP immunostaining. This is how astrocytes in an epileptic foci look from human patients. And we checked the methylation. The methylation was increased in these astrocytes in the epileptic foci. But methylation was not alone. Methylation was together in those astrocytes with reduced KIR4.1 immunostaining. So that means that these astrocytes cannot, buffer the level of potassium. So they induce neuronal hyperexcitability, which is basically the basis of epilepsy. And the glutamine synthase is also reduced in these, astrocytes compared with the normal astrocyte from a patient that died from another causes. So my conclusions are basically that initial precipitating injury, status epileptico, traumatic brain injury, etc., can induce a profuse rhabdogliosis, and this is together with astroglial DNA hypermethylation. And this is also observed in patients with drug-resistant epilepsy. So it seems to be a long-term situation in those astrocytes. This astroglial hypermethylation, it goes together with long-term downregulation of homeostatic genes, key homeostatic genes, together with an inflammatory response. And this, we call those astrocytes like epileptogenic astrocytes that facilitate epilepsy. Astroglial hypermethylation, mechanistically hypermethylation, seems to be induced by the pro-epileptogenic release of DAMPs like HMGB1, and the consequences is the repression of homeostatic genes. And the cytarabine could be an interesting treatment to prevent this effect. So strategies aimed to prevent astroglial hypermethylation during that silent period can have a profound impact in the development of the epileptogenesis, and we are in fact working in that direction. And my model, just to clarify if something is not clear, homeostatic astrocyte, happy astrocyte in the CNS, in the brain, suffer an initial precipitating event, could be trauma, status epilepticus, encephalitis, whatever, that induce a population of pro-epileptogenic astrocytes. These pro-epileptogenic astrocytes are obviously pro-inflammatory. This is known in the field. This has been known for a long time. But the new thing is that they also hypermethylate homeostatic genes, so they repress homeostatic genes. They are not so able to support neurons to survive, basically. This situation, combinated situation, increase the high epileptogenesis in this brain. And this is the people that work in this. This is the people of my lab and collaborators, but essentially this has been work of Dante, Alicia Rossi, and Paul that have been working really hard in this project. Thank you very much for your attention, and I will be here for questions.
Thank you very much, Alberto, for a very exciting talk. Now I'd like to welcome our second speaker, Dr. Kathryn Bowles. Dr. Kathryn Bowles completed her PhD at Cardiff University in the Professor Leslie Jones Lab. Then she moved to Icahn School of Medicine at Mount Sinai, New York, where she joined Professor Alison Gautier's lab as a postdoctoral fellow. She just started her lab in 2022 at the Edinburgh University in the UK Dementia Research Institute. She is working on tauopathies, and her lab is mainly interested in investigating MAPT expression, function, and splicing, and how these processes contribute to Alzheimer's disease, from frontotemporal dementia, Parkinson's disease, and other tauopathies. I'd like to thank you once again. Welcome, Kathryn.
Thank you very much for the introduction, Deborah, and I want to thank the organisers for inviting me to speak. I thought I'd talk a little bit about some of the work that we're doing in our lab to try and understand how communication between neurons and glia are altered in the context of tauopathies. So, ooh, will the slide change? Yes. So as mentioned, my group is interested in tau. We're pretty agnostic to disease as long as it's got something to do with the protein or the gene MAPT, or more broadly the genetic locus 17q21.31, then we're interested in it. So many people are familiar with tau in the context of Alzheimer's disease. It's the protein that makes up neurofibrillary tangles. We know that Alzheimer's disease causes a loss of memory and cognition, and there is a genetic association now between Alzheimer's risk and the tau locus. But there are a whole host of other neurodegenerative diseases that are all associated either pathologically or genetically with tau, of which this is just a snapshot of a few of them. So I'm not going to go through and tell you all of the details about all of these different tauopathies, but this is just to demonstrate to you that it's an incredibly heterogeneous group. They all use different forms of tau. Tau can aggregate in different brain regions, in different cell types, and that causes very varied clinical presentations and very different phenotypes. And what we really don't understand is how this one gene and this one protein can have such diverse neuropathological and clinical effects. However, what all of these tauopathies do have in common is they are neurodegenerative diseases. So they're all characterized by neuronal death. And so, of course, a lot of our interest has been typically on what's happening in neurons. So that's also supported by the neuropathology when you take a look at post-mortem brain tissues with people who've had these tauopathies. Neuronal pathologies are very common. This is an example of neurofibrillary tangles that you see in Alzheimer's disease, where you have these kind of characteristic flame-shaped tau inclusions. But in other tauopathies like frontotemporal dementia and Pick's disease, you can get some more varied pathologies like these kind of perinuclear horseshoe-shaped inclusions. You can get these balloon neurons. And over here we have these kind of globular spherical things called Pick bodies, which are characteristic of Pick's disease. However, in a huge bunch of these tauopathies, glial pathologies are also not only dominant, but they're neuropathologically diagnostic criteria. So for progressive supranuclear palsy, for example, you see accumulation of tau in the astrocytes. These are called astrocytic tufts. You also see astrocyte pathologies in things like corticobasal degeneration. This is an example of thorny astrocytes that are common in another disorder called RTAG. And you also see tau accumulation in oligodendrocytes. This is an example of something called an oligodendroglial coiled body. But even though there are these kind of dominant glial pathologies in some of these tauopathies, we really don't understand how this tau accumulation affects the function of the glia cell or how it subsequently affects the neurons that are surrounding it. And there's a pretty good reason for why people haven't really studied what's happening in the glia. And that's because tau is not really expressed very highly in the glia. So this is RNA-seq data from iPSC-derived neurons, astrocytes, and microglia. And you can really appreciate the vast majority of tau is being expressed by the neurons. And so it's really been thought for quite some time that the astrocytes and the microglia are just responding to whatever dysfunction is happening in the neurons, and they're not actively contributing to the disease process themselves. However, if you zoom in, there is, in fact, some tau expression in the astrocytes and the microglia. It's around 10,000-fold lower than what you see in the neurons, but it is there. And so if we just start off thinking about the astrocytes, we're starting to see an accumulation in the literature of some evidence that tau expression in astrocytes, endogenous tau expression in astrocytes, is influencing neuronal function and neuronal health. So there's this paper now from around 10 years ago where they cultured primary neurons from mice with primary astrocytes, and they were either wild-type neurons, that's the C57, or they were tau mutation neurons, which cause frontotemporal dementia. So that's this P301S. And so what they found is if you either had wild-type neurons or even mutant neurons, and you cultured those with wild-type astrocytes, the survival of those neurons was much better than if you gave the neurons a mutant astrocyte. So that's indicating that the genotype of the astrocyte and the tau that's endogenously being expressed by the astrocyte is having a direct impact on neuronal health. This has also been replicated in human disease models. So this is iPSC-derived neurons. This time they cultured wild-type neurons with tau mutation astrocytes. These are another mutation, IVS 10+16. And what they found is that if you gave those neurons a mutant astrocyte, you could increase this kind of hyperexcitability phenotype that's really characteristic of these tauopathy models. And just by reducing the tau in the astrocytes, leaving the neurons alone, you could rescue that phenotype. So again, the very low level of tau in the astrocytes is sufficient to modulate how the neurons are functioning. There's also some other work in the context of Alzheimer's disease, which is demonstrating you don't even need a direct interaction between the astrocytes and the neuron, but actually just conditioned media and the factors that astrocytes are excreting is sufficient to affect neuronal health. So in this context, they took primary astrocytes, they exposed them to A-beta oligomers, and then they put that conditioned media on neurons, and that caused neuritic changes and synaptic loss. But what was really neat is that when they knocked out tau just from the astrocytes, and then they repeated that experiment, this was no longer neurotoxic. So not only is tau modulating the way that astrocytes are able to interact with the neurons, but it's modulating the way in which the astrocytes are able to respond to pathologic stimuli around them, which then has a secondary effect on neuronal health. In our own hands, we have a lot of different iPSC lines that carry MAPT mutations. We focus a lot on this S305N. And when we grow those cells into astrocytes just by themselves in monoculture, we found that they have a pro-inflammatory phenotype. So we haven't done anything to them, we've not stimulated them, they're not grown with anything, just that endogenously expressed mutant tau is sufficient to increase the levels of this CXCL5, which is an inflammatory chemokine, in the mutant astrocytes. What we also found is that the genotype of the astrocytes influenced how they were able to respond to exogenous tau. So if the astrocytes were carrying one of these pathogenic mutations, they more readily ate up any exogenous tau that we added onto them. So that's demonstrated by the red fluorescence in these images. And these are just different tau isoforms, different forms of tau we've given to the astrocytes. And that was also true regardless of which isoform we happen to look at. You can kind of see it a bit better with this example, that the mutation astrocytes more readily accumulate and eat up that exogenous tau. So there's this really interesting interaction between endogenously expressed tau and how the astrocytes can respond to exogenously given tau. So next we wanted to know, well, okay, so we know that the astrocytes are performing differently because they have these tau mutations, but then how is that influencing neuronal health? So this was work done by Louis Kiwi, who's a PhD student in my lab. And what we did is we generated a mixed genotype model, the same as that kind of mouse model from a few years ago. So we could make wild-type neurons, and we cultured them either with mutant astrocytes or wild-type astrocytes, and then we did the opposite. So we took mutant neurons and we cultured them with either mutant astrocytes or wild-type astrocytes. And in my lab, we tend to sequence everything. So we collected these cultures and we did single-cell sequencing so we could pull apart what the transcriptomic changes were due to all these different kind of genotype and cell type combinations. And so this is just like a little highlight to show you that the astrocyte genotype was in fact sufficient to change neuronal transcriptomic expression. And so here, what this is showing you are wild-type neurons if they've been cultured with mutant astrocytes compared to wild-type astrocytes. And so that astrocyte genotype is causing disruption in all of these different signaling pathways. A few that we're particularly interested in is that we see the mutant astrocytes are reducing glutamate receptor binding, they're reducing vesicle-mediated transport, and they're reducing neuronal projections in those wild-type neurons. But we wanted to dig a bit more into how were neurons and astrocytes communicating with each other in this context. So we used a software called CellChat. And what this can do is it can estimate receptor-ligand signaling pairs based on the expression of the receptors and the ligand genes in those different cell types. And so what was really interesting that we saw is that when you gave wild-type neurons a mutant astrocyte, they had significantly more predicted receptor-ligand signaling pairs compared to the wild-type neurons with the wild-type astrocytes. So just the presence of that mutant astrocyte is changing the way in which these cells are able to communicate with each other. And so what was really neat is then if we look at all of the kind of incoming interactions and outgoing interactions for these different cell types, we can see the astrocyte genotype has a difference on the neurons. And so first of all, I have sequenced here the astrocytes cultured with mutant neurons. The neurons have a huge effect on the astrocytes as well. And then we have astrocytes here cultured with wild-type neurons. But what was really exciting to see is that here we've got mutant neurons cultured with the mutant astrocytes, and they've got this elevated level of incoming outgoing signaling compared to all the other neurons that we assayed in this experiment. But if we give those mutant neurons wild-type astrocytes, it brings their signaling and their communication way back down to exactly the same as a wild-type neuron. So it's just kind of highlighting the impact that the astrocyte genotype can have on regulating neuronal function. So that's just a little snapshot of what we're doing with astrocytes, but how about the microglia? Microglia are much more challenging to understand in the context of tauopathies. They don't have a pathology or sort of tau aggregation the same way that you see in astrocytes or in the oligodendrocytes. There's also genetic data that's the most recent GWAS from PSP, for example, shows no association of the genetic risk with microglia function whatsoever. If you compare that to Alzheimer's disease, there's a huge microglial component there, but for PSP, it's completely lacking. However, from human PET data, we can see that there is a really nice correlation between the amount of inflammation that's detected in the brain with the rate of progression of disease. And that actually looks very similar to the correlation between disease progression and tau accumulation measured by PET in the same people. So it's indicating that there is some, at least, predictive relationship between how microglia are functioning and how they're behaving and how tau is accumulating in the brain. There are also studies that have taken a look at microglia in vitro. So these are primary microglia, again, from mouse models. And if you expose them to tau, they phagocytose it very readily. But what's really interesting is if you then expose them to tau again later on, they're nowhere near as responsive. They don't pick it up in the same way as they did the first time round, like they've kind of become desensitized to it. And then what's really neat is then if you take a tau mutation microglia, it's even less responsive. So it's not able to respond to its environment in the same way because it's already been exposed to that kind of pathogenic endogenous tau. And there's also a recent study looking at in a human model. So these are iPSC microglia with that IPS10+16 mutation again, cultured by themselves, and they are impaired at phagocytosing whatever substrate gets put on them. So they're not able to eat up E. coli as quickly as wild-type microglia are able to do. And we do see that there are these transcriptomic changes caused by the tau mutation. So much like the astrocytes, it's demonstrating that even very low levels of tau expression are sufficient to modulate the function of these glial cells. And so we've taken a similar approach as we did for the astrocytes, where we put together these mixed genotype co-cultures between neurons and neurons and microglia. And the first thing that we did was in the monocultures, actually, we wanted to explore how the microglia might respond to synaptosomes. So one of the kind of overarching hypotheses at the moment in the literature is that in neurodegenerative diseases, microglia become hyperactive and they sort of aberrantly phagocytose all of these synapses and they engulf them, and that contributes to synaptic loss and then subsequently neuronal death. So we wanted to see if that was the case for these tau mutation microglia. So what we did was to isolate synaptosomes away from organoids that were either wild type or carried the same tau mutations, and then we could label them, put them on the microglia, and then track what happened. And so like the other paper saw with E. coli, we found that the tau mutation microglia were indeed impaired at phagocytosing these synaptosomes. But what was really interesting is that the wild type microglia in blue here were able to tell the difference between synaptosomes that contained pathogenic mutant tau, so that's these kind of white circles at the top, and wild type containing synaptosomes. But the mutant microglia were not really able to tell that difference, so these two black lines here. And so we single cell sequenced everything, much like we did with the astrocyte cultures, and we found that the microglia responded to a pathogenic environment differently depending on their own genotype. So much like the response to the synaptosomes. So what I'm showing you here are gene expression changes in either wild-type microglia or mutant microglia when they're cultured with mutant neurons compared to wild-type neurons. And so if you have a wild-type microglia in that pathogenic environment with a tau mutation neuron, you get upregulation of all these pathways you'd expect, like a response to stimulus, you get locomotion things, you get lots of things that are indicating that they're detecting something is wrong in the environment and they are putting out an appropriate response. However, when you have a mutant microglia, they fail to do that entirely. They're not responding to the environment appropriately, and instead they're just dying. So the microglia, even though they are responding to the neurons, their own genotype defines how they are responding to the neurons. But of course, we're also interested in how the glia are influencing the neuron's health. So we can do the opposite comparison. Here we now take the differentially expressed genes in the neurons, if they're either wild-type neurons or mutant neurons, if they're cultured with mutant microglia compared to wild-type microglia. And so wild-type neurons don't really care. There are really very few transcriptomic differences if you gave them a mutant microglia or if you gave them a wild-type microglia. But if you already have an impaired neuron, one that's got a tau mutation, then there's a really detrimental effect of also giving them a microglia with a tau mutation. And so we're seeing upregulation of pathways to do with oxidative stress and oxidative phosphorylation, lots of these metabolism changes that are consistent with increased levels of cell stress. So just having a microglia that's not able to support the neurons appropriately is progressing the disease mechanism in a way that a wild-type microglia wouldn't do. So the final thing that we did is to see whether the microglia genotype could influence neuronal firing. So not just gene expression changes, but functionally what they were doing. And so we did this using a multi-electrode array. And if we had mutant neurons with mutant microglia, we could replicate this hyperexcitability phenotype, which I mentioned earlier is very reproducible in these tau mutation models. If we give wild-type neurons a mutant microglia, like the transcriptomics, they don't care, it doesn't change anything. But if we give the mutant neurons a wild-type microglia, we can restore that signaling back down to normal levels. So overall, the point I'm trying to make, and hopefully have made over this talk, is that mutant tau can influence neuronal glial interactions really quite significantly. What's really quite surprising is that even these very, very low levels of tau expression is sufficient to alter cellular function. In terms of the astrocytes, we see that they're in this kind of pro-inflammatory state. It affects how they interact with tau that they come across exogenously, and it's severely impacting how astrocytes and neurons are able to communicate with each other. In terms of the microglia, we kind of see the opposite end of the spectrum. They actually fail to respond to toxic stimuli, and they're not able to properly regulate synaptic function. And so I think, what we find from this data is that it's really important to have both mixed and single cell systems. We see cell autonomous phenotypes in our single cell systems that are really helpful to understand and to study. But we then pick up these different phenotypes and these different interactions when we look at these mixed culture systems. And so just a little plug for the review that we have in this special issue. And we take a look at what's known in human model systems about neuroglial interactions in the context of different neurodegenerative diseases, so not just the tauopathies, that really kind of highlight how we need to dig more into how all these different cell types are communicating with each other, because ultimately it's all connected. And this is often me when I'm thinking about these projects. So thank you very much for your attention. Lots of people have kind of contributed to this data and can make the lab what it is, and also all of the funders and the patients who have donated their materials so we can make iPSC lines and look at their brains. Thank you.
Thank you, Kathryn, for your great talk. I'd like to now welcome questions. I have a few questions here, and if you have more questions, please send us your questions. So for the first speaker, Alberto, we have a few questions here. First of all, how does the prevalence of epilepsy vary across different parts of the world? Why do you think this is the cause?
Yeah. I mean, there's just speculations about that. The problem with the countries that are not fully developed is that usually there are many epilepsies that are due to injuries in the newborn kids. I mean, during the birth and in the following years due to infections and maybe not so good attention at the beginning. And there are some other situations. I've been in Peru last month in the meeting of epilepsy, and we know that the high mountains there are more epilepsy due to, we don't know why, because it's the diet, it's the high the lack of oxygen. We don't know, but the reality is that the number of patients in most of the countries are around the number I gave you. It's like 50 patients per 1,000 per 100,000 population, and that's basically the number. But the most of the patients are in not fully developed countries. That's the real situation today. Yeah.
There's another question for Alberto. Do you think the observed DNA hypermethylation in astrocyte is the cause of epileptogenesis, or is it a consequence of repeated seizure activity?
Yeah, could be both. We see that just one seizure, one long-term seizure in animals can induce hypermethylation, and then those astrocytes that are hypermethylated are not so good for neurons. They're pro-inflammatory and they don't support neurons, so they are not good astrocytes at all. But the reality is that we also see hypermethylation in the patients that have been years having seizures, and that's why they got surgery, because they cannot control the seizures with anti-seizure drugs. So the reality is that probably every seizure induce more hypermethylation, or at least they keep the hypermethylation like a signal, keep on going signal. And the other thing is that the promoters of DNA methyltransferases have NF-kappa B binding sites, and NF-kappa B is the main factor of neuroinflammation. is a trophic factor, the transcription factor of neuroinflammation. So probably there is a cycle there of methylation and NF-kappa B activation due to neuroinflammation and keep on going. So my answer is both things. Yeah.
So I have a question. You think inhibiting NF-kappa beta signaling could also be a way to prevent this?
Yeah, we tried that, and the results are not so good because the problem is that NF-kappa B is not just used for neuroinflammation. Neurons require NF-kappa B to survive, and not in epilepsy, but we tried to do it in models of sleep apnea, and the results were not good because a little bit of NF-kappa B is good. The problem is that when they get overactivated. So we tried that, and we couldn't get nice data in at least in those kind of cyclic diseases, because sleep apnea is quite similar. We have cycles of sleep apnea that they are accumulating the damage, and so probably NF-kappa B, inhibiting NF-kappa B in acute in neuroinflammation, encephalitis, for example, could be good. But for the long term, for sure, it's not good. At least in our hands, it wasn't good.
I have a question for Kathryn. Do you think that your iPS models would be ideal to study these problems, and why you think this would be ideal? Because some studies were done with different isolated cell types. How you think this could help us answer your questions?
So, I mean,
system.
So, I mean, iPSC models are, I mean, I'm obviously a fan. The lab is built on iPSC models, but I think there's a lot of questions they can answer. But I think it is also important to be aware of the limitations of iPSC models. So one of the strengths of them is that you're keeping a human genetic background. And when we're looking at things like glia, there are big differences between rodent models and in humans in the way that like astrocytes and microglia function. So the iPS are really useful for that. We also try and expand our studies to have as many genetic backgrounds as we can handle. So I think that can also be a bonus. In terms of your question, yes, they're really great for being able to mix and match in a way that's a bit more challenging perhaps than with some of the mouse models, which we leverage all the time. We also do a lot of organoid work so that we can look at how these different cells can communicate with each other and how they interact with each other. But we do also have to bear in mind that there's still just a model. They don't completely recapitulate what's happening in the brain. Lots of people criticize them because of aging. But yeah, the mix and match and the way you can kind of refine the model to exactly your question, I think is a huge advantage of the iPS.
So there's a question here. You think they would recapitulate more the phenotype seen in the brain?
Yeah, so, in the brain, none of the cells exist in isolation, right? So, we do neuronal monoculture models, but, there's only so long the neurons can survive because they need that metabolic support from the astrocytes. And we can see in astrocytes that, the maturity changes if you culture them with a neuron and vice versa. So there are some things that do stay the same regardless of the way that you model it. But we are getting closer to how the brain actually functions if you're increasing the complexity of your model and including these different cell types.
Okay, there is another question for Alberto. So you presented data in overexpression of epigenetic proteins, but they were wondering if the function of these proteins is altered as well. So, and if you did quantification from immunofluorescence images also.
Yeah, we quantify. I mean, all the data I showed is all quantified in immunohistochemistry. That's a key situation. That's usually we work in that way. And the other thing is that the answer that I could give you is that we didn't check every protein that we saw alterated because we don't believe in. I mean, I can check if the glutamate transport is altered in those the glutamine synthase, but the problem is that I don't know how is the rest of the situation. So what we did in one of the last papers of the lab was to check how is the conditioned media obtained from astrocytes, how interact with neurons. So the whole package. So conditioned media of astrocytes exposed to pro-epileptogenic molecules like HSV1, we use that conditioned media to treat neurons, naive neurons that never saw, a problem. And those astrocytes basically kill neurons. And when I inhibit DNA methyltransferases, they don't kill neurons, at least not in the same amount. So overall, the physiology of these astrocytes are changed due to HNGB1 pro-epileptogenic treatment. So we believe, I am biochemist, so I like to do, to analyze transport and, check, how the molecules move from one place to the other. But the problem is that here we have the whole package, because astrocytes are supporting neurons in many different ways. They provide antioxidants, they give lactate, they take out glutamate. So if I check that they don't enough lactate, it does mean that neurons are gonna die? No, because neurons can use glucose. But the situation that we saw is that when astrocytes are exposed to pro-epileptogenic molecules, they kill neurons. And if you inhibit DNA methyltransferases or NF-kappa B, as Deborah said in the last question, they don't kill neurons. So that's my justification for not doing the specific studies on each transport.
So we'd like to wrap up. I'd like to thank everyone for attending, and especially the speakers for such great talks, exciting work going on that I will follow on. And I'd like to invite you to continue talking in the Biochemical Society website, or Blue Sky, or Instagram, LinkedIn. And again, I'd like to mention that this webinar is celebrating the upcoming issue, Imbalance of Homeostasis in the Nervous System. So I'm still welcoming you to submit articles, reviews about keeping homeostasis, aging, epigenetic modifications, metabolism, and other studies. So visit our website again, and feel free to suggest other topics for this series of webinars or other speakers. And thank you very much. And finally, if you want to see other webinar series in the YouTube, you can see other webinar series, and this one will be there as well in a couple of days. So finally, I want to highlight how it is important to be connected with scientists working in the biomolecular area. And so become a member of the Biochemical Society, which makes this possible. There are some courses featured, also career development related. And it's important for us to connect internationally with people from all over the world. Visit our website. Thank you once again.